My name is Armen Ovsepian from Institute Pasteur, and I would like to present you our work with the title Cumulative Core Genome MLST Schemes for Increased Discrimination of Nested Phylogenetic Groups. Core genome multilocus sequence typing. It's a widely used and powerful method for bacterial strain genotyping. However, it entails inherently a trade-off between its discriminatory power and the breadth of applicability, since the size of the core genome decreases as the phylogenetic breadth of target group decreases. So to address this, we have devised a cumulative cgMLST approach. So to do this, for any bacterial isolate, any bacterium, we could take genome assemblies and then identify the core genomes at different phylogenetic depths. And then we will use these core genomes to develop schemes for different phylogenetic depths with no redundancy for the loci between the schemes. And after the integration of these schemes in a database, we could combine these schemes depending on the isolate of under investigation to make high-resolution genotyping. So in our case, in the study, we have implemented the cumulative cgMLST approach to Klebsiella pneumonia, for which core genome MLST exists for the species complex, but is restricted to only 629 loci. So in this study, we have developed a core genome MLST for Klebsiella pneumonia sensu stricto. And this scheme contains extra 2,759 loci. And in combination with species complex loci, this covers more than 60% of the total protein coding sequences of Klebsiella pneumonia. Additionally, we have developed also schemes for the important sublineages of Klebsiella pneumonia, sublineage 147 and sublineage SL307. And these schemes, loci of these schemes in combination with the more generic schemes for species complex and Klebsiella pneumonia sensu stricto, they cover more than 83% of total protein coding sequences of Klebsiella pneumonia. So to test the performance of a cumulative cgMLST strategy for these sublineages, SL147 and 307, we tested their performance in outbreak datasets, and we could see in these figures. And we could see that the pairwise distances obtained with cumulative cgMLST analysis were very similar to the distances obtained with single nucleotide polymorphism analysis, yielding Pearson correlation coefficient of 0.97 for the case of SL147 and 0.93 in the case of SL307. And so we see that they perform very well. And so as any method, it has its limitations. In this case, the main bottleneck of cumulative cgMLST analysis would be the development step of the schemes, which can be time-consuming. And to ameliorate this and accelerate this step, we could use automated bioinformatic pipelines, and which is something we are already working on it. And by doing so, we could apply cumulative cgMLST on existing phylogroups or even emerging phylogroups, and this can offer us a very robust, rapid, user-friendly, and very sensitive tool for bacterial genotyping. With that said, I would like to thank, of course, all my colleagues from Institute Pasteur. And I would like to say that this study was part and it's funded from the Centaur project, and it's funded by the European Union.